Assalamu alaikum everyone. I'm Abdul Razzak Chalab Cham. I am a third year medical student, and today I'm going to be explaining two lectures, the spermatogenesis lecture and the sperm defects lecture. These two lectures are very short and very simple. Inshallah, they won't take much of our time. But before getting started, if you have any question, please feel free to contact me through my contacts here, either through email or WhatsApp. I will be more than happy to help you with any of your questions, inshallah, especially that this is a pre-recorded question session so that it won't be much interactive. These are the objectives of the lecture, very short and very simple. Let's get started. So we'll start off with a quick revision over mitosis and meiosis. I know that you have done this like 1 million times by now in high school and in uni, but we will try to get over it quickly. So we know that mitosis consists of one step, one division, while meiosis consists of two cell divisions. This is the main difference between them. Mitosis starts with a 2N cell, which basically means that it has 46 chromosomes, or 23 pairs, and these chromosomes are going to replicate, and then during metaphase of mitosis, these chromosomes are going to align in the middle of the cells, getting ready for the cell to divide. And when the cell divides, it's going to produce two daughter cells. As you can see here, the way the chromosomes are aligned, we know that chromosomes consist of two sister chromatids, okay? So this is one sister chromatid, and this is one sister chromatid. And as you can see here, by the way these chromatids are aligned, chromosomes are aligned, when the cell divides, the two sister chromatids are going to be separated from each other. So one cell is going to get this sister chromatid, and the other one is going to get the other one. But this way of dividing, or this way of division, is going to cause that the two daughter cells are going to have the exact same amount of genetic material, okay? What about in meiosis? So in meiosis, we also start with a 2N cell. The chromosomes are going to replicate, but then the chromosomes are going to align during metaphase 1. We said that meiosis consists of two divisions, meiosis 1 and meiosis 2. So during metaphase 1 of meiosis 1, okay, the chromosomes are also going to align in the middle. But as you can see here, the way they are aligned in meiosis is different than the way they are aligned in mitosis. In mitosis, each chromosome is aligned individually so that when the cell divides, sister chromatids are separated from each other. However, in meiosis 1, the chromosomes are aligned in pairs, in homologous pairs, which means that one chromosome is from the father and one chromosome is from the mother, okay? The way they are aligned here is that when the cell divides, these two chromosomes are separated from each other, and each one of the daughter cells gets one total complete chromosome, okay? Either the paternal one or the maternal one. If we assume the green one is the paternal one and the blue one is the maternal one, we can see that after division, one cell will get the paternal and the other one will get the maternal, for sake of simplification only. Now, however, there is a twist. Not all the chromosome is not totally paternal or not totally maternal, because while the chromosomes are aligned in the middle there during metaphase 1, waiting for the cell to divide, they're going to have a small talk, okay? Let's assume this is a maternal chromosome, and let's get another color here for the paternal chromosome, okay? This is a paternal chromosome. While the two chromosomes are aligned in the middle there and waiting for the cell to divide, they're going to exchange some genetic information, okay? So the maternal chromosome is going to get some paternal genes, while the, let me change the color again, while the paternal chromosome is going to get some of the maternal genes. This exchange of genetic information between the two chromosomes is called crossing over. This is a very important process that helps in the genetic variation of the offspring, okay? Because it helps in increasing the variation, okay? So again, during metaphase 1 of meiosis, the two chromosomes align in the middle, homologous chromosomes. They have some exchange in information called crossing over, and then the cell divides, separating these homologous chromosomes from each other. And we get these daughter cells of meiosis 1. However, these cells are still not ready for fertilization. They have to undergo one more division so that they are ready. And this is meiosis 2. So in meiosis 2, these chromosomes again align in the middle, and the cell divides, producing also two daughter cells. But this time, just like mitosis, the sister chromatids are going to be divided from each other. Okay. By the end of meiosis, we're going to have four daughter cells that are completely different from each other. Why are they completely different from each other? That's because of the crossing over process. And having different daughter cells is going to cause more variation in the children. Okay. Now, a very important thing that you need to know is that sometimes not everything goes as planned, and sometimes we have some mistakes that happen. Sometimes some chromosomes fail to separate from each other. But when everything happens as normal and everything happens as it should happen, we call that correct disjunction. In other words, separation happened correctly. Okay. And when that happens, everything goes well, and all the daughter cells have the same amount of chromosomes, which is about 23 chromosomes. Not about, which is 23 chromosomes. Okay. However, when sometimes separation of chromosomes fails to happen, we call this non-disjunction. In other words, separation did not happen. Okay. And this non-disjunction could happen either in meiosis 1 and in meiosis 2. And in both cases, it's going to produce a case of aneuploidy. Aneuploidy basically means that one or more chromosomes are not diploid, which means that one or more chromosomes or one or more cells have an extra copy of a chromosome or a less or a missing copy of a chromosome. Let's see that on the picture again. Okay, so let's assume that non-disjunction happened in meiosis 1. So we see that here is the two homologous chromosomes should be separated from each other. But here they fail to separate, and both homologous chromosomes go to one side. Okay. And so we can see that this cell now have an extra copy of a chromosome. Okay. While this cell have a missing copy of a chromosome. So these are aneuploid cells. Okay. And when meiosis 2 happens to these two cells, we are going to have a result of 4 aneuploid cells that are either having an extra copy of a chromosome, like these ones, or have a missing copy of a chromosome, like these ones. Okay? This is in case of meiosis 1. What about in meiosis 2? In meiosis 2, the first meiosis 1 happened normally, and all the cells now have an equal amount, but this time, instead of sister chromatids separating correctly during meiosis 2, they both go to the same cell. So now we have a cell with an extra copy, while we have another cell with a missing copy. Okay. Don't really have to worry about these numbers, number of aneuploid cells, number of abnormal cells. But what you have to know is that when there is non-disjunction, either in meiosis 1 or in meiosis 2, this is going to result in aneuploidy. Okay. These abnormal cells or abnormal daughter cells, if they happen to fertilize or get fertilized, in 60% of cases, they are going to produce spontaneous abortion. So, aslan pregnancy is not going to happen firsthand. Okay. However, in the remaining 40% of cases, okay, they're going to produce some syndromes, like the famous Down syndrome, where you have a trisomy 21. Trisomy 21 basically means that the instead of having 2 chromosomes at chromosome 21, you have 3. Okay, and you have an extra copy of this. So this is trisomy 21 or Down syndrome, or such as Klinefelter syndrome. We know that it's normal to have either an XX for a female or an XY for a male. Okay, but in case of Klinefelter syndrome, we have six chromosomes instead of 2, X, X, Y, and this is going to cause Klinefelter syndrome. Okay. Another one that you guys already studied, I guess, in the previous lecture is Turner syndrome, which is X0, which basically means that you have only one female chromosome, but the other one is missing. There is nothing. This is the case where there is a missing chromosome, not an extra one. Okay. So what you need to know from this slide is that if correct separation of chromosomes happen, we call this correct disjunction. When there's a problem with separation, either in meiosis 1 or meiosis 2, we call this non-disjunction. And this is going to cause aneuploidy. Aneuploidy, in 60% of cases, if fertilization happened to an aneuploid cell, is going to cause spontaneous abortion in 60%. and 40% is going to cause some syndromes related to the number of chromosomes, either an extra copy or a missing copy. Now, before digging into information about spermatogenesis, let's discuss some information. Let's try to separate it into multiple steps. Okay. So the first step of spermatogenesis is actually mitosis. We have these cells called spermatogonia. Okay, and these cells continue undergoing mitosis continuously. Okay, they keep undergoing mitosis so that they produce even more spermatogonia. So what basically happens is that these spermatogonia keep undergoing mitosis, producing more spermatogonia. We're going to discuss more in the next slide, okay? But some of these spermatogonia start to differentiate into these cells called spermatocytes. And these spermatocytes are the first part of the following step, which is meiosis, okay? So next step is meiosis, and we just discussed that meiosis have two steps. Meiosis 1, meiosis 2. So we start meiosis with a primary spermatocyte. This primary spermatocyte, which is originally a spermatogonia that differentiated into a spermatocyte, okay? This primary spermatocyte will undergo meiosis 1 and produce something called secondary spermatocytes, two secondary spermatocytes. Then these secondary spermatocytes are going to undergo meiosis 2, and they're going to produce these spermatids. As you can see here, spermatids look nothing like the normal sperms, okay? So they have to undergo more modifications to become like similar to the actual sperms that we know, okay? This takes us to the following step, which is spermiogenesis. Spermiogenesis is basically the differentiation or the modification of these spermatids that look like normal cells into these sperm-looking cells, okay, which are called sperms, okay? The final step of spermatogenesis is spermiation, which is basically the release of the sperms from the Sertoli cells into the seminiferous tubules. It's a very short step and very easy to understand, okay? All these steps take about 64 to 72 days to produce sperm. So the whole process of spermatogenesis from mitosis to spermiation take about 64 to 72 days, okay? This is very basic, and now we're going to go into more details. Let's start off with the first step, which is mitosis of spermatogonia. So we have this cell called spermatogonia, and these are basically stem cells, and these are diploid cells, which means that they are 2N. They have 46 chromosomes, okay? These cells are located in the basal compartment of the blood testis barrier. So before moving on, I'm going to discuss a little bit about the blood testis barrier, okay? So this is a simple drawing I drew. This is a lumen of the seminiferous tubule. These are two Sertoli cells, and this is the bloodstream, okay? It's a very simple picture, only to help you understand, okay? So between these Sertoli cells, we have these adhering or tight junctions, okay? These tight junctions. And these tight junctions function to separate the sperms that are located in the seminiferous tubule from the bloodstream. Why would you want to separate the sperms from the bloodstream? You basically don't want these sperms to get exposed to the immune system there. Because if the immune system saw these sperms, it's going to attack them, because it's going to identify them as a foreign body, okay, as a foreign object. It's going to attack these sperms, and it's going to kill them all. And this is going to lead to infertility. You don't want infertility, therefore, the function of this blood testis barrier is to separate these sperms that are in the lumen of the seminiferous tubules from the bloodstream. The blood testis barrier consists of two compartments, the basal compartment here, okay, and the abdominal compartment here. Abdominal compartment, from the name, is adherent to the lumen, okay? The basal compartment is at the base. You can remember this by remembering that bloodstream and basal are close to each other, B with B, and abdominal is basically close to the lumen, so it's the one closer to the lumen, okay? Now let's go back to the previous slide and discuss more about mitosis of spermatogonia. So we have these spermatogonia there in the basal compartment of the blood testis barrier, which is separated by these tight junctions from the abdominal compartment. These spermatogonia will keep undergoing mitosis and to ensure that you have a good amount of stem cells to produce sperms. If these spermatogonia stop undergoing mitosis, after a while, they're all going to be used and you will have no more stem cells to produce sperms. That's why these spermatogonia have to keep undergoing mitosis so that they ensure that the body has a good amount of stem cells to produce sperms. And this is going to maintain the ability of the testes to continuously produce sperm at any time. Okay. However, some of these spermatogonia start to differentiate into primary spermatocytes and after multiple divisions. Okay. These spermatocytes are then going to jump over the blood testis barrier from the basal compartment to the abdominal compartment, and this is going to be the first step of the meiosis step. Okay, so again, first step of spermatogenesis is mitosis. We have these differentiating primary spermatocytes that are going to jump over the blood testis barrier, and they're going to jump to the abdominal compartment, and they're going to start undergoing the next step, which is meiosis. Okay. So second step is meiosis of the primary spermatocytes. So these primary spermatocytes, which were originally spermatogonia, will undergo the first step of meiosis, which is the meiosis 1. So they divide from primary spermatocytes to secondary spermatocytes. Simple, okay? So primary spermatocytes undergo meiosis 1 and become called secondary spermatocytes. Then these secondary spermatocytes will further divide with meiosis 2, and they're going to produce things called, or cells called, spermatids. These are the spermatids, okay? These are spermatids, okay? These spermatids look nothing like the sperms, and they look more like the spermatogonia than they look like sperms. So they have to undergo more another step to actually look like the sperms that we know, okay? And this step is spermiogenesis. So spermiogenesis basically happens between Sertoli cells here. You can see that these sperms or spermatids are inside the between the Sertoli cells, and they are going to undergoing modifications. These modifications include condensation of the nuclear material, formation of the acrosome, elongation of the tail and formation of the tail, mitochondrial spiral formation, and the removal of the extra cytoplasm. Okay, I'm going to discuss the significance of all of these modifications because all of these are important for fertilization later. Okay, now let's try to correlate each part of the sperm with its function and its role in fertilization. The first one is the head, okay? So the head basically contains the nucleus, which contains all the genetic information that the sperm is going to carry. A sperm without genetic information is useless. It has nothing to do. Even if it fertilized, it's not going to produce any fertilized egg. Why? Because the genetic information includes the XY genes and all the other genes that are needed for the baby to develop. Okay, so without these genes, you're going, there's no even if fertilization happens, the sperm is going to be useless because it has no genetic information. Okay, the second part is an acrosome. Okay, and it's basically a vesicle that contains degrading enzymes. Why would you need degrading enzyme in sperm? Basically, you have oocytes, okay, or eggs, okay, and around these eggs we have this area or this coating called zona pellucida. This is a protein coating that sperms usually cannot go through, but with the help of these acrosomal enzymes or degrading enzymes, the sperm can break through this zona pellucida and reach to the oocyte. So again, this acrosome or acrosomal vesicle contains this degrading enzyme like hyaluronidase and acrosin, which are going to break through the zona pellucida and help the sperm reach the oocyte or the ovum to fertilize it. Okay, without these acrosomal enzymes, the sperm cannot penetrate through the zona pellucida. It's impossible. Okay. The third part is the tail, and it's very simple. The function of the tail is sperm motility or sperm mobility, movement of the sperm. The last but not least part is the midpiece. Okay, the midpiece is basically consisting of spiral mitochondria. This spiral mitochondria is going to provide all the energy that the cell is going to that the tail is going to need to move. Without the energy provided by the mitochondria, the tail is not going to be able to move. So as you can see here, during the process of spermiogenesis, the spermatids undergo all these modifications to look like this sperm. But one very important modification that we have to know is the reorganization of the cytoplasm. So basically, you have an excess cytoplasm here in the spermatid, and during spermiogenesis, the cell is going to shed off this excess cytoplasm. Doesn't need it. It's going to cause problems. We're going to discuss more about these problems in the following lecture. But for now, you need these spermatids to get rid of this excess cytoplasm in order to look like the sperm that we know. Okay, this sperm. So this is basically all what I said in a written way. We have reorganization of the nucleus, development and positioning of the acrosome from Golgi apparatus. This is the very important thing, is that the origin of the acrosome is actually the Golgi apparatus. Okay, we have the assembly of the tail and the reorganization of the cytoplasm. Okay, now after spermiogenesis has occurred, and we said that spermiogenesis actually happens where between Sertoli cells. Now, the next step is going to be the release of these sperms from the Sertoli cells into the seminiferous tubules. And this is actually the last step of spermatogenesis, which is spermiation. Spermiation is basically the release of these mature sperms from between the Sertoli cells into the seminiferous tubules. This table here includes multiple events that happen during spermiogenesis, right? Yeah. And this is some of their functional events. I've already discussed them, but I will go over them quickly again. So we have nuclear chromatin condensation. We said that this is very important because they have the genetic information about the 6 chromosomes, X or Y, boy or a girl. Okay. We have the development of the acrosome, which is very important, which has the enzymes that are going to help in penetrating through the zona pellucida and help in fertilization. We have the development of and the growth of the flagellum, which is the main function is motility through the female genital tract. And we have the formation of the mitochondrial sheet around the flagellum. And this mitochondria is going to provide the ATP, fructose-derived ATP, for the flagella to move. Okay. Now we're done with spermatogenesis. We're going to discuss some of the functions of these Sertoli cells. Okay. Another name for these Sertoli cells is actually nursing cells. And as the name indicates, they have a supportive function. What do they support? They support the developing sperms or spermatids. Okay. They maintain and break and form junctions with the developing sperms. And the main function, or a very important function, of Sertoli cells is maintaining the blood-testis barrier. If you are going to forget everything about Sertoli cells, forget it, but remember this very important thing, is the maintaining of the blood-testis barrier. Okay, because without it, all the sperms are going to die. They also have a function in phagocytosis. They also transfer nutrients from other substances from the blood to the sperms. If we come back here to this image, we can see that these sperms are very far from the bloodstream. Okay, so they need these Sertoli cells to take these nutrients from the bloodstream and provide them with these nutrients so that they can develop well. Okay, and the last thing is the development of paracrine factors. Sertoli cells also have exocrine functions. These include the production of the fluid to move immobile sperms out of the testes toward epididymis, because immobile sperms cannot move by themselves. So they need some sort of fluid to help them move. Another very important function of Sertoli cells is the production of androgen binding protein. This protein is very important because it's going to bind testosterone. And you know, guys, that testosterone is very important for the development of sperms. It has to be present inside the lumen of the seminiferous tubule. So this androgen binding protein is going to bind testosterone, and it's going to increase its concentration a lot inside the seminiferous tubule, so that the sperms are going to use it to develop. Okay, if this testosterone was not present there in a high concentration, the sperms won't develop well. The last one is spermia. The last chronic function of Sertoli cells is spermiation that we have already discussed, which is basically the release of these sperms into the seminiferous tubule. The Sertoli cells also have endocrine functions. They express receptors for androgen receptors and follicle stimulating hormones, basically testosterone and FSH. Why would you need these receptors? They need these receptors because they help them in helping the sperms modify and develop. And also, when these hormones increase in the bloodstream, these Sertoli cells are going to sense it, and they're going to produce this hormone called inhibin. This inhibin hormone goes to the pituitary gland, and it tells it to stop secreting any more FSH. Why does it tell it to do so? Because there is a high amount of FSH. So high amount of FSH is going to be sensed by the Sertoli cells. Sertoli cells are going to produce inhibin in reverse to inhibit the process of the production of FSH. Also, Sertoli cells have a very important function, which is anti-Mullerian hormone production. I think already discussed that. It basically prevents the development of the female genitalia, of the internal female genitalia. And last but not least, is aromatization of testosterone to estradiol 17B. This has a local effect, and it's not really that much endocrine. Now, last concept of this lecture is actually decapacitation and capacitation. I'm going to explain capacitation first. So basically, capacitation happens inside the female genital tract, specifically in the oviduct. Okay, and it's basically capacitation is basically a series of modifications that the sperm undergoes so that it helps it fertilize the eggs. These modifications include stuff like an increased movement speed, increased like survival in acidic environments like the uterus, and also the acrosomal reaction. However, you don't want these modifications to happen inside the male genitalia. Why? Because for two reasons. Because it's useless, because there is no egg to fertilize inside the male genitalia, and because after capacitation, it takes only 4 to 6 hours for the sperm to die. And you don't want the sperm to die already inside the male genitalia. So you want some sort of process to delay this capacitation process until the sperm reaches to the egg, okay? And this is done by decapacitation. So decapacitation is basically a reversible process that's going to make the sperm less active. It's going to make it less capacitated, okay? Or incapacitated, or in another word, inactivated, okay? So it's basically a series of interactions between these proteins that are secreted by the accessory glands and the epididymis. They're going to interact with the sperm surface proteins, and they're going to delay the onset of capacitation. Until when? Until these sperms are released and they reach the oviduct. In the oviduct, this decapacitation process, we said, is reversible. So now it's going to be reversed, and we have capacitation activated. When capacitation happens, we have a series of modifications such as an altered membrane fluidity, which is going to help the sperm live in the acidic environment of the uterus. Also, removal of glycoproteins. These glycoproteins, when they are removed, this is going to allow the sperm to attach to the zona pellucida and start the acrosomal reaction with the help of the influx of calcium. Also, for phosphorylation of numerous proteins. You're going to discuss these more in later lectures that are related to fertilization, I believe. So if you don't really understand what is happening here, just keep them on top of your mind, so that when you take the fertilization, more information about fertilization, you are going to relate these ideas with each other. Okay. Now we're done with the first lecture, which is spermatogenesis, and we're going to discuss some sperm defects. This is a shorter lecture. I copied most of the slides from Dr. Abdul Jabbar's lecture for this lecture because there's not really much to add to it. What Dr. Abdul Jabbar wants you to know from this slide is that there are multiple causes for infertility, multiple causes for male infertility in specific, such as testicular insufficiencies, varicocele, low sperm count, and some are unknown reason. Idiopathic means unknown reason. That's it about this slide. You don't really have to worry about the numbers, obviously. Next thing, what's a normal spermatozoa? So we just discussed the process of spermatogenesis, and we discussed the normal shape of the sperms. So we said that it has a head that has an acrosome that breaks through the zona pellucida. It has a nucleus that contains genetic information. It has middle piece that has the mitochondria that's going to provide the energy, and it has a tail to move. However, there's something to add in this slide. We have this protein that is originally released by the glands inside the sperm, by the male genital system, okay? Specifically by the epididymis and the accessory glands. So we have this protein called glycodylan, okay? This glycodylan protein somehow has an ability to bind these abnormal sperms more than the normal sperms, okay? So if you have this normal sperm, okay, and let's say this is a very abnormal sperm, okay, it has the ability to bind this abnormal sperm more than this normal sperm. And in general, the function of this protein is to suppress capacitation, okay? What's the significance of this protein? So let's imagine that now the sperms are inside the female genitalia, and they want to go through the cervical mucus, okay? They need capacitation to start at that point. However, these abnormal proteins have a lot of glycodylan attached to them, and this glycodylan is going to prevent them from undergoing capacitation. Therefore, this glycodylan is some sort of mechanism to prevent these abnormal spermatozoa from undergoing capacitation and reach to the egg and fertilize it, and only allow the normal spermatozoa to undergo capacitation, go through the cervical mucus, and reach to the egg. I hope this is clear. This is clear. Okay. Now we have some abnormal changes that sperms usually undergo, or that sperms can have, that can have, like changes in chromatin condensation, defects in acrosome reaction, problems in tail motility. Obviously, is going to cause problem with motion and movement. Increased apoptosis or necrosis. This basically means increased sperm death. Okay, and some genetic abnormalities. There's a very important term that Dr. Abdul used to explain in previous years. I did not read it in your transcription though, but I think I might mention it just for the sake of completion, and you might, there is a slight chance that you get asked about it, and it is azoospermia. Okay, azoospermia is basically when someone can't produce any sperm, like zero or a very low amount of sperms. Azoospermia is basically when someone cannot produce any sperms. Just keep this on top of your mind. You might get asked about it, but as long as Dr. Abdul not mentioned, just don't worry about it a lot. Okay. Now, this is a slide that you can skip. It's basically just like an index to show how much of an abnormality does a patient have, how much of abnormal sperms does the patient have. Don't worry about this, even doctor said to skip it. This slide shows some abnormal morphologies of sperms, such as giant, micro, double head, double body, long head, rough head, abnormal middle piece. I have never seen any questions about these in the past, but you might want to read them like once or twice and forget about them. Even the name is very indicative of the shape. So even if you get somehow asked about them, it's not going to be a little bit a lot of struggle. Okay. But usually they don't get asked. I never seen any question about them. But this case of globosospermia is very important. This globosospermia means that the sperm has a balloon on a string morphology. A globe means a ball, and this can help you relate the ball to a stick morphology or a balloon on a stick morphology. This is very important because globosospermia is going to cause defective fertilization. Defective fertilization, in other words, is going to cause male infertility. Now we're going to discuss some sperm head deformities or abnormalities. There's not really much to explain here. I'm just going to try to simplify as much as possible. The first one is a microcephalic head. Okay, as the name indicates, cephalic is head and micro is small. So this is a small head, and it's usually associated with an acrosome abnormality. I'm going to discuss more about acrosome abnormality on the following slide. We also have total globosospermia. Globosospermia is basically linked to abnormal Golgi apparatus during spermatogenesis. We said that Golgi apparatus is the thing that condenses and it forms the acrosomes. So if there is an abnormal Golgi apparatus, you're going to have a problem with the acrosomal reaction. And this is globosospermia is usually associated with male infertility, okay? Most of the time. Another abnormality is basically thin or elongated heads. It's highly associated with hyperthermia for 6 to 24-month duration, okay? Hyperthermia is basically an increase in the temperature. I don't know if you studied this yet or not, but the location of the testes inside the scrotum is very significant for sperm development because it's somehow located outside the body. And it's very important for the sperms to have a lower temperature than the body's temperature. Because if there was an increase in the temperature, these sperms are going to struggle to develop and they're going to develop some abnormalities. So the location of the testes inside the scrotum, outside the body, is very significant for these sperms to have normal shape. So if you have hyperthermia for some reason, okay, there is going to be an increase in the amount of sperm deformities, especially thin and elongated heads. A very important cause of hyperthermia is varicocele. Very, very important and very, very high yield. There is no interview question without a varicocele question, okay? Just note, okay? The last abnormality is macrocephaly, which basically means a larger head. And it's associated with multiple deformities over the sperm, like an acrosome, multiple tails, and abnormal head. It's also highly associated with high rates of aneuploidy and polyploidy. Only know the name, no need to worry about this, what polyploidy, but aneuploidy, we just discussed it, one or more chromosomes are not diploid. Reduced acrosomal reaction. So we said that around the oocyte or around the egg, there is a protein coat that's called the zona pellucida. And there are specified enzymes that are located in the acrosomal vesicles that help the sperm break through it and reach the egg to fertilize the egg, okay? So if for some reason we have a reduced acrosomal reaction or reduced function of these enzymes, the sperm is not going to be able to penetrate through the zona pellucida and reach the oocyte, and therefore filtration is not going to happen. Okay. Okay. I believe we're going to learn more about zona pellucida in the following lectures with the female physiology. Okay. Another important abnormality is the residual cytoplasmic material. We said that during spermiogenesis, we have this cytoplasm shedding off from the spermatids so that they get the shape of the sperm. However, it's not only like shape-wise, there is a very significant reason for the cytoplasm to be removed or to be shed off. Basically, there is a small amount of cytoplasm that should be there that's helpful in both hyperactivation, incapacitation, and acrosomal reaction. However, if you have an excess amount of cytoplasm, if not all the cytoplasm was shed off, we're going to have cytoplasmic enzymes that are going to produce reactive oxygen species. I believe from your mole course last semester or last year, you guys took a lot about reactive oxygen species. But if you don't remember, these are basically very dangerous molecules that are going to cause damage to multiple cell organelles, including the DNA, mitochondria, and other stuff. So you really don't want them to be produced. But if you have an excess in cytoplasm, these cytoplasmic enzymes are going to produce a lot of reactive oxygen species and be harmful to the cell. Okay? Okay. Now we will discuss more about tail abnormalities. Okay. Let me go up this. Okay. So tail abnormalities. We know that the function of the tail is very, very significant and is very, very important for sperm motility. So that if there are any abnormalities in the tail, is going to cause a decrease in motility. And the flashy word for that for the decrease in motility is actually called asphenozoospermia. Okay, this is just like a very like luxurious word just to say a decrease in motility. Okay, and most of the time, or in many cases, these defects in cilia, these defects in the tail are usually associated with congenital defects in the cilia. We know that the structure of the cilia and the tail are very close to each other, so that sometimes you will have a combined chronic expiratory infection with the male sterility due to a congenital defect of the cilia. Okay. The abnormalities of cells could vary. They could be absent, they could be angular, they could be double, they could be they would come in many colors and shapes, and all of them are going to have a negative effect on the sperm motility. The effect of temperature, we discussed the effect of hypothermia, which is basically temperature. This is just only this slide is only just to tell you that some research was done on some medical students where they had a febrile event, such as fever, okay, they were sick. And after that, the research showed that they have a decrease in the amount of normal sperms, which basically means, in other words, that the amount of abnormal sperms was increased. That all of that was due to an increase in the temperature, okay? Testicular cancer. Cancer can never be good, so you would expect all types of deformities to form due to cancer. This includes microcephalic spermatozoa, post-acrosomal region abnormalities, acrosomal defects, excessive residual cytoplasm, and short tails. Again, it can cause multiple abnormalities in the sperms. A very high yield concept is varicocele, okay? And varicocele is just basically the pooling of blood inside the veins or the testicular veins, okay, which are taking blood away from the testes. We know that in all veins we have these valves. Let's assume this is a vein, and these are valves, okay. These valves prevent the backflow of blood toward downward, okay, or backward. However, in case of varicocele, these valves are dysfunctional, okay, and this is going to cause blood or allow blood to go back and pool in the veins. And blood is very much hotter than the environment in the scrotum. So a small amount of blood is okay, but when you have this much of blood inside the scrotum, this is going to cause hyperthermia to the scrotum and to the developing sperms, and this is going to cause multiple defects in the sperm parameters, either low, altered, or defective sperm parameters, like tail dysfunctions, or we just talked about thin and elongated heads. So varicocele is very important, and it's very common, about 10 to 15%, and it's actually the #1 cause of male infertility in the world, okay? Last idea of this lecture is chemotaxis. So let's say these sperms were deposited inside the female genital tract, and now they want to reach to the egg, okay? How would they reach it? They have three methods or three ways to reach it, or three helping methods to reach it. The first one is thermotaxis, the second one is muscle contractions, and the third one is chemotaxis. The main and the most important one is chemotaxis, but we will discuss thermo and muscle contraction first. So thermotaxis basically means that the sperm is going to detect the high temperature and is going to follow it. So as you can see here, as you go closer toward the egg, the temperature is going to increase, okay? The sperm are going to detect this increase in temperature and just follow the heat, okay? Another way is muscle contraction. You have contraction in multiple organs of the female genital tract, just as the cervix, the vagina, the uterus, okay, and even the oviduct, okay. So these muscle contractions are going to help guide the sperms toward the eggs. But the most helpful way for the sperms to reach the egg is actually chemotaxis. Chemotaxis basically means that the sperms are going to detect some hormones released by the eggs, such as progesterone, and other hormones that you don't need to worry about. But for now, sperms are going to detect this increase in progesterone in this area, and it's going to go there. Obviously, any sperm that can detect the heat well, or can detect the progesterone well, or even cannot move well, are going either to stay here or go to the opposite side or in random directions. Okay, that's it for these two lectures. I have a couple of questions for you to answer before we end the session. Question 1 says that which of the following cells undergo meiotic division? What which of these cells undergo meiotic division? Primordial germ cells, primary spermatocytes, and secondary spermatocytes, or spermatids? Feel free to pause this video and answer this question like as slow as you want. So the answer here is primary spermatocytes. Okay, the primary spermatocytes are actually the first step, the first cell that undergoes the meiotic division. Secondary spermatocytes might be there, and it might be a little bit confusing, but I don't think it's going to be both in the exam. I think you might be able to appeal it. This is the graph that I just explained during the lecture. Primary spermatocyte undergo first meiosis, or meiosis 1, produce a secondary spermatocyte. Secondary spermatocyte is going to undergo meiosis 2 and produce a spermatid. Okay, then these spermatids are going to undergo spermiogenesis. Okay, to produce sperms. This is a graph that I made by myself. Just like a very brief summary for spermatogenesis. Spermatogonium is going to undergo meiosis and produces even more spermatogonia, and then is going to differentiate. This is differentiation here into primary spermatocyte, undergo meiosis 1, produce secondary spermatocyte, then which are going to undergo meiosis 2 and produce spermatids. Okay, and then these spermatids are going to undergo spermiogenesis and become sperms. Question 2. When in spermatogenesis does crossing over of the chromosome happen? During mitosis, during meiosis 1, during meiosis 2, and upon fertilization. So basically, upon fertilization is totally wrong because خلاص يعني it's too late. During mitosis, also we discussed in mitosis, there is no crossing over that happens, and all the cells that are produced are identical. So it's either meiosis 1 or meiosis 2, and this is a simple recall question, and the answer is B. Okay? Again, during meiosis or prophase 1 of meiosis, when the cells are aligned beside each other before the cell divides, they're going to have some exchange of genetic information, and then they are going to divide into two daughter cells. Okay. Question 3. Which of the following seminal plasma proteins play a role in the suppression of capacitation? Okay. This is very important, and it's very specific detail of the lecture. And the answer is, this is a recall question, and the answer is glycodylan. We said that glycodylan is going to bind to the abnormal sperms, and is going to prevent them from undergoing capacitation, so that when they reach the mucous membrane inside the female genital tract, they won't be able to undergo capacitation and go further and fertilize the egg. Last question of this lecture is actually is the image. Oh, I actually missed the image. The image shows an abnormal sperm with Globosospermia. Okay, I'm going to edit it and add it in the PowerPoint. But for now, Globosospermia, remember it, the balloon shape with a tail. Okay, a balloon on a stick shape. Okay. What could be the cause behind this abnormality? Either a residual cytoplasmic material, failure of chromatin condensation, problems with tail motility, or abnormal Golgi apparatus. The answer is also a recall question, and the answer is abnormal Golgi apparatus. Okay, guys, so for this lecture, it's mostly memorization. I try to simplify everything as much as possible. I hope everything was clear and everything was easy. So these are again my contacts. If you have any questions, feel free to contact me at any time. And please don't forget to scan this code. Please scan the code. It's only literally one question that's going to take like 30 seconds max, like even less than 30 seconds. So please scan this code, and thank you guys for watching. Inshallah, I will see you later, inshallah.